Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar titled Pioneering the Development of Point-of-Care CAR T-Cell Therapy Manufacturing in Canada, Experience from the CLIC-01 CD19 CAR T Clinical Trial. I'm Caitlin Crouch, and I'll be moderating today's webinar. And joining me today is Dr. Natasha Kekre, who will explore the foundational work that was needed to launch the pioneering Canadian-led Immunotherapies in Cancer Research Group, which offers CAR T-cell therapy to patients across Canada. After the presentation today, we'll have a live Q&A session where we invite our audience to pose their questions to Natasha using the Ask a Question box at the bottom of the screen, and we'll try to get to them during today's session. Now, I'd of course like to introduce today's presenter. Dr. Natasha Kekre is a hematologist within the Transplant and Cellular Therapy program at the Ottawa Hospital. She is also an associate scientist within the Ottawa Hospital Research Institute and associate professor of medicine at the University of Ottawa. She completed her bachelor's in science before obtaining her medical degree from the University of Ottawa, where she then trained in internal medicine and hematology. She then went on to do a fellowship in stem cell transplantation at the Dana-Farber Cancer Institute in Boston, Massachusetts, with a master in public health from Harvard University. So without further ado, it gives me great pleasure to hand over to Natasha to kick us off with today's presentation.
Thank you so much for having me. Today, I'll be presenting about our Canadian experience with point-of-care manufacturing for CAR T-cell therapy. This is the experience of the CLIC group, the Canadian-led Immunotherapies in Cancer group. My name is Natasha Kekre. I'm a hematologist in Ottawa, Canada, and thank you again for attending. So today's objectives, I want to go back in time to 2017 to sort of tell you what the landscape of CAR T-cells looked like in Canada at the time. And elsewhere in the world, of course. Talk a little bit about how we came up with this Canadian CAR T manufacturing platform, the research progress in our current platform, as well as the future of this platform and where I think CAR T research is going in Canada right now. So to start with, we can't forget about the original CAR T-cell trials for CD19 CAR T-cells, which has been the most studied and well reported in the literature. CAR T-cells, very briefly, is a T-cell that's been taken from a patient and re-engineered to express an anti-CD19 chimeric antigen receptor on it, which allows it to attack CD19 on tumor cells. And so that was kind of the initial experience in the marketed world of CAR T-cells, has been based on really these two pivotal trials, which showed that patients with relapsed and refractory acute lymphoblastic leukemia and certain types of lymphomas could indeed respond very well, despite the fact that they had very aggressive tumors that had really run out of standard of care options. We were seeing response rates in the 80% range, and we were seeing survival at a year of 60% or higher, and that was really quite remarkable. And so this, of course, led to the approvals of a few products in the US and has led to many approvals now in the US and in the world for CAR T therapy. And that really was back in 2017. That was the landscape we were seeing of these approvals happening in the US and wondering when and how this might reach Canada. But quite frankly, in Canada in 2017, there was really no access to these life-saving therapies. We knew that the growth of these things was very intense and fast because we were seeing products be approved when there was not that many patients treated. It was quite quick, as we know. And most patients in Canada could not access these products, whether it was through commercial access or clinical trials. So the way it worked in Canada at the time is that a select number of patients, when these products were approved, were able to get to the US to be treated. But this meant leaving your home, leaving your family, moving to the US for at least 1 month, if not longer. And oftentimes these patients were just much too sick to get to the American trials. A few of these clinical trials did indeed open in Canada, but they opened in 1 or 2 sites in Canada with very limited spots. We're talking about a few patients per year that were allowed in these sites. So really, this was not an option for Canadians. And lastly, if a Canadian patient wanted to go to the US to access a clinical trial spot, they would have to have $1 million American in cash before they could even be considered for the consult, let alone to actually get to these clinical trials. So that was incredibly prohibitive for our patients here in Canada. And so people like me who are hematologists and see these patients knew that we had to do better. We had to create a system where we could have CAR T-cell clinical care and clinical products, but also a research platform where we could make our own CAR T products or make the next generation of CAR T products. And, we have great scientists who can work on these innovative medicines, but we had to make sure we were finding a pathway to reach our Canadian patients. And so we knew that we needed access to the therapy. We also needed access to clinical trials. And we just really in Canada could not wait on big pharma companies to reach Canada for these products. And so many of us met back in 2017 at a conference called the Cancer Summit. This was hosted by BioCanRx. This is the Biotherapeutics for Cancer Treatment. This is a nationally or federally funded entity that funds research and grants for enabling translation of cancer therapeutics to the clinic, which is really what this group endeavored to do at the time. These were all of the people who I'll name in a minute who were involved in the program, but these were people from across Canada and different research institutions, most notably the Ottawa Hospital, where I am based, and BC Cancer, where many of my colleagues were based, who came together to create sort of a Canadian platform for CAR T therapy. And so we really went on the premise that, okay, we know that these products exist. We know that they have shown great results in the landscape of clinical research, but we didn't want to start from scratch with a new concept, a new design, and build the preclinical development needed to reach a clinical trial. That process in itself, just like it was for CD19 CAR T cells, that process can take over a decade. And so instead, we wanted to work from, well, can we actually produce a clinically effective product entirely within our Canadian system that would benefit our Canadian patients? And so we wanted to work towards GMP manufacturing of the vector, the virus, the CAR T cells, make sure that we had all the quality control measures in place for release testing, and then actually reach a clinical trial, which we thought would be a more effective way to build this platform than to start from a brand new CAR T target and design that would require many, many years to finish the preclinical development. And so that's what we did. We built a program centered around a CD19 CAR T product where we had centralized vector manufacturing. This was led by Robert Holt in Vancouver, British Columbia, which is on the west coast of Canada. We had the virus being manufactured in Ottawa, which is on the east coast, towards the east coast of Canada, with Dr. John Bell, who led that, and he has a clinical grade manufacturing platform. We then had Dr. Brad Nelson, again on the west coast in Victoria, British Columbia, who actually knew how to manufacture T cells and ran our point of care cell processing and manufacturing for this Canadian-made CAR T platform. And we slowly built the infrastructure within Canada. We had trainees and technicians and physicians involved across the Canadian ecosystem to ensure that we could build jobs and innovation within this platform in Canada. And ultimately, that platform, that project was to build access for Canadians to clinical trials for CAR T cells. Within that, because we're Canadian, we really also wanted to make sure we were doing this the right way for Canadians in the long term. So we built a second program called Go-kart, which is getting better outcomes from CAR T therapy. And the purpose of Go-kart was really to ensure that we were thinking of the barriers to implementation in Canada so that we were not dealing with the same barriers and challenges that CAR T programs that were already built had already encountered. We wanted to learn the lessons of what had already been done, not start from scratch. And so that was a really important part of our platform, was to include this second arm that really focused on the implementation science of getting this to our patients. So to give you again more detail about this first project, the CLIC program, like I said, Victoria and Vancouver and British Columbia, this is where the vector was made by Rob Holt, and the CAR T cell production was done by Brad Nelson. The virus production was way over here in Ottawa with John Bell. And then myself, and back then Raywin Brody, we were in charge of finding patients in both sites and collecting T cells, and of course, doing the CAR T infusion and the clinical care that would be needed for these patients. And this was, although the team is much bigger now with many, many more people, this was the original people who built the clinical trial, along with many other people, but really this was how we got started. And so our second project was Go-kart, like I said, and I wanted to run through what that looked like. So part A of this was to build sort of a systematic review arm that could look at all the preclinical and clinical data that was available for CAR T cells so that we were not reinventing the wheel, but instead learning from the literature that was out there. We then had part B, which was really the knowledge translation. That included patient partner, Stu Schwartz, as well as a physician partner, Matt Seftel, who basically would help us identify barriers and facilitators to making this happen for patients. So this involved a lot of outreach led by Justin Presso, really to show what would be the barriers to patients reaching the trial, what would be the barriers to clinicians referring patients to the trial. Because in Canada, we are fraught with the unique problem, which is that we are a huge country with very limited centers that can do CAR T or stem cell transplant therapies. And therefore, we wanted to make sure that Canadian barriers and facilitators to this research were being thought of before we built our trial. And so that was the knowledge translation arm. We then built an economic arm led by Kednapa Tavorn, really looking at what are the cost effectiveness parts of this that we want to look at when we build our program. Is our program going to be cost effective? What does cost effectiveness for CAR T cells even mean in this day and age, knowing the price tag attached to commercial products? How could we do this in our clinical trials and analyze it properly? And so we had, that was our third arm. The next arm, which was led by me and Raywin, was really to look at a cohort study to test our criteria for the trial and kind of come up with a very realistic number of accrual numbers, beds that would be required in hospital, and the resources that go into a big, expensive trial like a CAR T cell trial. And that was really the importance of this arm, was to gather data on how to make sure we're ready for these patients to come into hospital for the clinical trial. And that was the fourth arm. And finally, we wanted to do some testing, which was very unique to CAR T cells, about how to make sure the products can get from one side of the country to the other. How do we make sure that we are manufacturing in a timely fashion, that patients are getting to the products, to the referrals, all that stuff in a timely fashion. So the last arm was really a test run of the manufacturing procedures that we were creating. And so that was the program. CLIC was the name of our trial, Canadian-led immunotherapies for cancer. And Go-kart was basically our strategic planning for the trial. And so here was our manufacturing strategy. So Miltenyi has, of course, this Clinimax Prodigy machine that we thought would be the best in terms of implementing this manufacturing across Canada, because it's a closed system, it's easy to operate, it's easy to implement in different centers. And so we thought this would be our best platform to start with. So I already told you that we had a plan for making the transgene and the lentivirus in centralized way, but the goal with Dr. Nelson's group was to have these prodigies in multiple centers across the country so that we could manufacture these CAR T cells in a point of care manner. And we relied on a very well-established 12-day manufacturing protocol that Miltenyi already had, and already had multiple trials that were enrolling patients using a similar manufacturing platform that we could, again, build on instead of starting from scratch. And so this was our timeline. We basically came together around 2017, got some funding from this BioCanRx group that enabled us to build the package needed for Health Canada, which is our regulatory body here in Canada. We then had to make sure we had the prodigies up and running. We had all of the quality control measures in place to manufacture the CAR T cells. And then we had to do our dry runs to really prove to Health Canada that we could do this in multiple ways. So we did it over 3 manufacturing runs to show them that we could meet our specifications for product release. And so in 2019, we received what's entitled our no objection letter from Health Canada to go ahead with our trial. We then obtained ethics approval. And then, of course, there was a delay in getting contracts because this program was led by the Ottawa Hospital Research Institute as well as the BC Cancer Research Institute. These are 2 separate academic institutes, but this was a collaborative endeavor. So we wanted to ensure that there was mutual liability and mutual understanding that both groups were responsible for this trial and for the outcomes and for manufacturing. And so that is a unique way of doing a trial, and that took a lot more time and energy than I had expected, so that lawyers were happy at both institutions, that both were liable for parts they should be liable for, but could also share in the liability for the trial. And so that part is a little bit unique, and that is one of the barriers to getting us open, was really getting those contracts in place. But I was happy that by October, we were able to open our clinical trial in 2019. I do like to show this slide, not to show all of the pitfalls we had, but that in opening any clinical trial that requires biomanufacturing, such as CAR T cells, there's always going to be hurdles that you have to overcome. And so the red here shows that this is just the first year of our program. The green is sort of all the things that were working the way we expected them to work. But the red shows times where we had problems. We had a contamination at one point. At one point, the place that makes the virus, that the titer of virus was not what we expected. And so we were worried we didn't have enough virus to go ahead with the trial. And so these were all kinds of little hurdles we had over time with manufacturing that we had to overcome to eventually reach clinical trial. But this was part of what it is to manufacture under a Health Canada regulated program, just like it would have been in any other country that's opening a CAR T-cell clinical trial like this. It's a learning curve. It does take time to overcome these barriers in order to reach a clinical trial. But we did overcome those barriers and eventually opened our CLIC-01 trial, which is an autologous anti-CD19 CAR product for the treatment of relapsed refractory CD19 positive hematologic malignancies. This includes really any CD19 positive malignancy. So ALL being the most common, non-Hodgkin's lymphoma like DLBCL and mantle cell lymphoma were sort of the next most common indications. But we included Richter's transformations, follicular lymphomas, indolent lymphomas, CLL, really any disease that was relapsed or refractory and who could potentially benefit from a CD19 CAR. And so first we tested safety in the first 20 patients, and then we opened it up more broadly to test efficacy in another 80 patients. And I will say this was a fresh product, so it was delivered fresh no matter where in Canada the product was collected. It was delivered fresh to British Columbia. It was manufactured, and 12 days later, it was sent back fresh to the site that would infuse the product, and it was given back to patients. So around day 8 of the manufacturing protocol, we had to have a go, no-go from our manufacturing sites to start the flu-cy lymphodepletion chemotherapy so that we could go ahead and get the patient ready for CAR T infusion. And so we've now published the first 30 patients worth of data. We've actually treated now over 80 patients, but the first 30 is public information. And we had patients who, we had some patients with higher performance statuses, so ECOG of 2, which would have been uncommon in the pharmaceutical trials. We had a range of diagnoses, DLBCL still being the most common, and mantle cell being the second most common. And then you can see the range of disease stage in the LDH at the time of CAR T. In terms of prior therapies, we had a range of 2 to 6, 3 therapies being most common prior to getting to this trial. A majority of patients were primary refractory or had a very early relapse, which would be expected for this. We had a handful of patients with a previous allo transplant and a previous auto transplant. 2 patients had actually had both. And here was the results of our manufacturing. Without going through all of the numbers, our manufacturing went quite well. We made a surplus of cells. The median dose infused, which we did have a dose reduction for patients with a significant bulk of tumor. We had a 10-fold dose reduction for those patients. But outside of that, our median dose infused was 2.55 times 10 to the 6 cells per kilogram. And all of our patients except one reached the max total dose of cells to be infused because, like I said, with the Prodigy, we did make quite a surplus of cells, I would say. We did have 2 early manufacturing failures very early into starting the trial. One was that we shipped the cells at ambient temperature because that is what the company that we used for our healthy donor apheresis products was using ambient shipping before we ever used patient products. We know that shipping at 4 degrees is probably better from a cell viability point of view and manufacturing, and so we did change that protocol at the time of that manufacturing failure. The other failure was thought to be related to seeding the Prodigy with too few cells, so this was increased from 50 E6 to 100 E6 on the Prodigy. And since that time, in these first 30 patients, we really had no manufacturing failures. This remains true. We've had only 2 other manufacturing failures, but both have been deemed related to the patient cells, not the manufacturing itself. And so in terms of feasibility, of course, we opened in October 2019 and then COVID hit. Luckily, our trial was considered, life-saving potential for many patients, and therefore it was still given priority to run when we could. But there was a 3-month pause in recruitment due to COVID. Outside of that, I think our numbers look quite good in terms of study screening to enrollment of 18 days. But most importantly, apheresis to CAR T infusion is 15 days. And we also, we do have access to commercial products in Canada that is significantly longer than that 15-day turnaround. So for these very sick patients, we really were excited to show that we were able to get patients to CAR T infusion very quickly from their collection date. And in Canada, this is really important because patients often come a long way to get to the treatment center. And if they come for apheresis and they can be treated within 2 weeks, they often don't have to then fly home or drive far to get home, only to then come back to our center. Of course, we're in Canada, so we have to worry about weather delays and snow and things like that. And so for us, this is actually really important from a patient perspective, not just a physician perspective, because it allows patients to very quickly come in for apheresis and then get to the CAR T infusion. So in terms of safety, this was not really that surprising to us. We had CRS, as you would expect, and neurotoxicity, as you would expect. We did have 2 non-relapse related deaths. These were, in fact, the same 2 patients who did have high-grade ICANS or CRS. One was a primary refractory DLBCL, who, in fact, the CRS and the ICANS did resolve, but the patient had significant lymphoma in their bowel lining. And although the lymphoma on PET scan was better with the CAR T infusion, the patient had a very difficult bowel perforation and passed away quite quickly after a bowel perforation. So that still counts as a non-relapse death, despite the fact that I don't think it was related to the CAR T toxicity, although it probably was related to the CAR T treatment. The other patient had significant burden of acute lymphoblastic leukemia, had grade 1 CRS right after cells, had fever on the first day, but did progress to grade 3 CRS and had significant neurotoxicity, as well as multi-organ failure, and ultimately passed away on day 9. This patient had a marrow that was completely full of blasts and significant tumor burden, both in the marrow and extramedullary disease. So this was a patient we might have predicted to have a high risk of toxicity, but this was a patient who, was willing to still try to get to clinical trial, but unfortunately did pass away. In terms of other toxicities, really nothing that surprised me in terms of chemo-related toxicities or effect of CAR T therapy. Nothing really unusual here. We did see almost all patients with an undetectable CD19 level at day 28 and month 3 after the CAR T cells. One patient did have a transient time where the CD19 was positive, but that eventually also disappeared from the blood, and that patient remains in remission as well. So we saw what you would expect to see with CD19 CAR T cells. We saw those things even in our trial. And so in terms of outcomes, we had what we think were quite good results. We had responses. Many of the patients who had early partial responses, you'll see, did have complete responses eventually. So you can see we had some patients who transformed from partial to full responses. We did also have other patients who did not respond. That's the purple here, who progressed in terms of their disease. But you can see from this swimmer's plot that really there was more green than purple, and we were seeing quite a few responses in our trial. And this led to a median progression-free survival of these 30 patients of 6 months, which would be very much in keeping with the early phase 1 and phase 2 trials of the now approved pharmaceutical company products. And so I think this is quite reassuring. And the part that is really lost in this is that these are patients who were probably sicker than the patients who ever reached Zuma 1 or Eliana or any of those trials. And the reason for that is that these patients, didn't have the time to wait 4 to 6 weeks for manufacturing for pharmaceutical products, or they were too sick to be considered for pharmaceutical products. And so I actually think our cohort is sicker than what is out there for commercial products. And so seeing a PFS of 6 months in that cohort, to me, I think is really important to note, because these were very sick patients who had very little time left. We did divide the data, although it's very hard to make any conclusions at only 30 patients, but we did see really great outcomes in our ALL population. For the most part, everyone who was treated for ALL has responded for the most part. So that has been a very exciting story, and that is what we are looking forward to updating with our 80 patient cohort that we now have. And survival was 11 months median survival for this small cohort. So that was our CLIC trial that we built. But I also wanted to take some time to talk about Go-kart, this strategic planning that we did as well, only because I think it's really important as we build CAR T trials that are including this point of care, that we think about how to make these trials better in the future and designed better, especially in places like Canada, where it is hard to access pharmaceutical-driven trials, We have to spend this money very carefully that we get from granting agencies to make sure we're getting the most bang for our buck and really making sure we're getting the most out of these trials. So part A, I told you, of Go-kart was really to do a clinical review, and that's what this is. We published our CD19 CAR T review, looking at all the trials that have been published. What did the data look like? How did the patients respond? What was the patient criteria to get into those trials? And we published all of this as a very thorough overview at the time of the landscape of CAR T cells. And the purpose of this wasn't to show you the data that we published, but really to show you that we were able to come up with event rates for cytokine release syndrome and neurotoxicity that could guide how to power our trial and how to create stopping rules for our trial that were in keeping with what the literature already looked like. And so we used all of this for our regulatory package for Health Canada to justify the design that we created for our clinical trial. And to ensure that really, again, we were doing things based on what was already out there, not starting from scratch. So the next part of our Go-kart platform was really to look at knowledge translation. So again, we wanted to identify barriers and facilitators to building a CAR T therapy trial in Canada. We interviewed hematologists across Canada looking for what the perception would be of a clinical trial that was of a manufactured product in Canada. Participants were at least familiar with CAR T therapy, but at the time probably had very little knowledge of the evidence behind toxicities and efficacy. So what we heard from physicians is that they were motivated to screen patients, especially if they found that the screening was very feasible and easy for them to fit into their clinical load, but that the barrier would be adding time to their day to enroll or screen patients, resources needed to do that, forgetting that the trial existed, worrying about wait times to get to the trial if patients were very sick or already needing some palliative therapies to make them to give them time before saying goodbye to their families. And so we heard a lot of barriers about getting to the trial from across sites, especially for sites that were not located in Ottawa and Vancouver, where the patients were physically being treated for the trial. So this is super important to us because we want to make sure that we can break down some of those barriers when we build a clinical trial. And this led to a lot of outreach to physicians outside of Ottawa and Vancouver when we went ahead and built our clinical trial to ensure that we were helping meet some of these barriers and concerns before we found out that we could not accrue enough patients for our trial, for example. We did the same thing with patients. We interviewed patients across Canada. Again, they were motivated by understanding the benefits to the community, but also wanted to acknowledge that they prioritized their personal benefit and health if this was a trial that they could benefit from. We heard about the value that they had in receiving medical advice from their own hematologists and oncologists. They didn't necessarily want to go and see a physician in another city about this trial if that physician wasn't their own. They wanted to first be able to hear about the trial from their own doctors. And again, this created outreach for us to make sure that physicians were comfortable discussing what CAR T cells are before they ever referred them to the trial. And patients did talk about very practical things like making sure they had their expenses covered, especially if they're traveling from far, making sure the caregivers were supported, things that we were happy to include in our trial design based on what we heard from patients across the country. And again, I recommend everyone do this when they're building clinical trials. This was incredibly valuable in building not just patient partners, but building things into our trial that would help to facilitate the trial design and enrollment. Next, we had part C was really the economic modeling, which to me is really important because when the price tag attached to CAR T cells is in the six-figure range, when comparing, to products that we know on the market are more than $400,000 American, it's really important in a publicly funded healthcare system like we have here in Canada that we acknowledge, how do we make this better in the future, and how do we ensure that the CAR T program we're building is actually better economically for our patients in Canada, which is a huge consideration for countries like Canada. And so our objective with this was really to look first at the therapeutic headroom and something called the maximum reimbursable price for CAR T cells compared to alternative standard of care. And we have seen with some of what the companies have done with cost effectiveness models is they've built models that compare CAR T cells to palliative chemotherapy. Well, palliative chemotherapy is incredibly cheap. It's for an incredibly short duration. And that's not standard of care in this day and age for ALL or lymphomas. We have things like checkpoint inhibitors. We have fights out there. We have new antibodies. And so we really wanted to make sure we were making this model relevant in the current landscape of therapies that exist. And so this is just an example of one of the models we built for ALL that includes drugs like blinatumomab and inotuzumab, as well as transplant, all of which are standard of care therapy options for our patients with relapsed refractory ALL. And so we built a model where they could have any of these therapies in any different order, and then put CAR T cells into the model to see where would CAR T cells fit in terms of therapeutic headroom. Where can you get the maximum economic benefit for life years gained in a model like this? And that really was the purpose here. And we incorporated CAR T cells at different stages of this model to see how does it really impact the overall health, but the overall resources for these patients. And we came up with some different numbers. Again, using comparators that we think are very much in the real world now, WTP being our willingness to pay threshold. And interestingly, despite the fact that cancer therapies have gone up significantly in price, our willingness to pay threshold in each country or each place remains pretty much what it was many decades ago. So if we assume our willingness to pay threshold is about $100,000 per quality of life year gained, essentially you can see here what their costing looks like when you're comparing the maximum, what we would consider the best maximum reimbursement price for those therapies. And these were the reason I'm showing this is not to show you what the costing was of CAR T, but the maximum reimbursement price that would be acceptable when you're comparing to blinatumomab, you're comparing to inotuzumab, or you're comparing to chemo. Now, chemo is incredibly cheap, so the price that you'd be willing to pay for CAR T is much different than if you're comparing to more expensive drugs. But even then, the price of CAR T is still significantly less than what we know the marketed price is of CAR T cells in this landscape of CD19 CAR T. So I think it's really important not to know the numbers here, but to really understand how this impacts the CAR T therapy trials that we build and what kind of a maximum reimbursement price we need to be thinking about in the future. And so that was part C, and I don't want to give away all the results from that because Kednap Tavorn is publishing a lot of this information so that it's out there publicly available in terms of what the numbers look like and what we think an acceptable cost of CAR T would be moving forward, not so much what we're already being forced to pay right now. So lastly, we built a cohort study to look at numbers. And so we just wanted to look at, if we just looked at every ALL treated in Ottawa and Vancouver, how many of them right now would be eligible for our CD19 CAR T cells, and how many patients would we need to consider for these trials moving forward? So out of 173 patients at our two institutions, the number of patients who would be eligible for our CAR T product would have been 35%. So there's lots of interesting numbers here, the time from diagnosis to transplant, the overall survival of these patients, the time from the most recent relapse to death, because again, this gives you a time frame of how much time do you have to get these patients to trial. It's not long. These patients pass away within 3 months of their most recent relapse. It also gives you a sense of how many beds you need in the hospital. So about a third of your population of ALL were eligible for our trial. So we knew that moving forward, even if we opened another center to treat patients, we would be telling them, okay, look at how many ALLs you treat in a year. Of those, about 35% of them could be eligible for the trial, so that you can build realistic accrual numbers for trials like this. Because again, when each patient is so expensive and requires so many resources, it's really important to plan for that. And so that's what we did with this cohort study. So we built a clinical trial. We built manufacturing at the time, was only in Victoria, BC under Dr. Nelson. And we built this strategic plan called Go-kart to really help us to strategically open this trial in Canada. And now we're at the stage of building this platform across Canada so that we can actually call it point of care manufacturing. Because right now we are, even though we're using multiple Prodigy machines, they're all housed in Victoria, BC, which is out here. But we want it to be housed all across Canada so that you can manufacture the cells easily in multiple locations. And that is where we are right now. We are now setting up in Toronto and Ottawa and Winnipeg, as well as hopefully other centers across Canada, to build a pan-Canadian network of these Prodigy machines so that we ultimately can be a point of care manufacturing center. And this is being built with the help of our BioCanRx funders. And this is really to show this is 100% Canadian made. This, we think, over time would lower the costs. I didn't go into what the costs are yet because We want to be able to show in a lot of detail what the costing was like to build this platform, not just the cost of each patient product being made right now, which is a very simple, small number, much, much less than people would expect. But we have to account for the fact that we also built a lot of infrastructure and a lot of the base work that we did to house the quality control measures, for example. All of that work has to be accounted for in the costing. So it's not just simple to say, I know how much the reagents cost on the Miltenyi machine, I know how much we paid to make the virus, but there's so much more that we want to understand about the costing as we move forward. We think that this point of care network across Canada decreases the timelines of getting to CAR T therapy and, of course, leads to faster patient delivery. I already showed that we can get patients from apheresis to CAR T infusion of 2 weeks, which we think is quite good. And that is allowed by this point of care manufacturing system. The Miltenyi Prodigy allows us to do that. And if the treatment is at each site, then we can have standardized protocols across the country so that we are not relying on one center. So that, if there's a power outage or a massive storm or any, real world event that stops one center from being able to give the CAR T or to manufacture them, we can rely on other centers across the country. It also means hopefully less transport of cells. If you have multiple centers across Canada that can manufacture the cells, you don't have to always ship the cells all the way from this side of Canada to the other side of Canada, which is time and money, of course, to do that. So that's really the platform that we're now building after this trial is opened. We've proven our weight in terms of showing that we can adequately give these products and that they are effective. And now we need to roll it out across Canada. And I wanted to spend a few minutes talking about what that looks like across Canada. So to build this Canadian point of care network, we identified manufacturing facilities across Canada with criteria that we thought were important. We wanted them to be affiliated with a transplant unit or somewhere that's experienced in giving CAR T therapy. At least in Canada, CAR T therapy is still given by transplant wards and stem cell transplant programs. We wanted to make sure that they had some experience in clinical grade manufacturing of cell therapy products, because that is its own whole expertise in terms of quality of care, release assays, all of that stuff is really important in manufacturing. We wanted them to have some experience. They, of course, needed to have a Clinimax Prodigy machine. They had to have a willingness to embrace this collaborative trans-Canadian program. It needs to be a group who would not want to compete with us, but would want to work with us. And that's really the people we were looking for. And of course, we wanted them to be motivated to be part of this network and build up the next clinical trials in terms of cell therapies in Canada. And so this is how we approached building this network. And it was led by Brad Nelson and Rob Holt in British Columbia, who I told you were on the west coast of Canada, who did all of the manufacturing for the vector and the cells and ran our quality program. They led this buildup of CAR T manufacturing in other cities across Canada in this way. So they started with a gap assessment performed by that manufacturing center in BC to assess the level of equivalence or appropriateness of a site-specific technology. So things that existed in the site already, looking for potential regulatory gaps, things that make their program different. Because while each program could be very good at doing CAR T manufacturing, it is not the same as being equivalent to the CAR T manufacturing we were already doing in British Columbia. And to be part of one clinical trial protocol and one clinical trial package for Health Canada, we have to show the similarities between our programs and that we can effectively make the same type of CAR T product. We don't need to show that independently they can make manufactured products. And so that's a very important distinction, because this gap assessment allows for that BC site to confirm that another site can meet the same technology and manufacturing processes that we already have in place, because that is what we require to show Health Canada that we have other centers now being onboarded for the trial. We then perform a technology transfer package from the British Columbia site to other sites in Canada, again, to ensure that they follow the same processes we're already doing. And lastly, an audit performed by CFIL, that's the Conconi Family Immunotherapy Lab, that's the center in British Columbia, that's their title. They then send quality and manufacturing personnel to these other sites for an on-site audit to ensure that the point of care manufacturing site that we are adding into our network is ready to go to manufacture on this trial. And so how do we ensure that we are making a standard or consistent product across multiple sites? I'm sure there's many different opinions on this or how to do this, but for us in Canada, in our group, we decided the split batch runs was the best way. Take a healthy donor apheresis product, split it in half, and manufacture at the same time in 2 different centers simultaneously a CAR-T product on the Miltenyi Prodigy machine. We also plan to do site-by-site comparability studies when multiple sites are enrolling patients and treating them at different centers with products manufactured at different centers. We want to do a site-by-site comparability, but that's going to take a lot of time because to be powered to do those comparisons, you have to treat a lot of patients. And so that's not something that we can do overnight. Instead, overnight, we could compare the sites by manufacturing benchmarks and show that all of our products meet a basic benchmark that we will outline for Health Canada to show that all of us are following the same criteria for manufacturing. And this remains exceedingly rare, even with these Miltenyi prodigies around the world being used to do phase 1, phase 2 clinical trials for CAR-T. There are not many trials in the world that we know of that have multiple manufacturing sites with prodigies in different places under the same clinical trial number, under the same regulatory requirements of one clinical trial. We've seen some great data comparing a similar CAR-T product and platform in Russia and the US, for example, but these were under 2 separate clinical trial numbers, 2 separate regulatory bodies. They were not combined into one. So that's what makes this different, is we want this under the umbrella of one clinical trial. And so that's where we really have to meet a different level of standard. And so our BioCanRx CLIC platform, this is ultimately what we think will happen, is that the cost of decentralizing our CAR-T production in this academic, not-for-profit setting that our hospitals work in will eventually lead to lower costs, because that fixed cost that I talked about, that is very high at the beginning for building the infrastructure, building up the trials, is very high at the beginning. But this goes down with time. And so ultimately what we're trying to do is improve the fixed cost, but the variable cost, of course, will stay the same. And over time, this will lead to an ultimate lower cost per product. And especially as more products are made each year, this will also come down with time. We also hope with time we will avoid shipping delays, we'll decrease our vein-to-vein time, we'll improve access to these products for these patients who have very rapidly progressive disease. And overall, because this is an automated Miltenyi Prodigy manufacturing platform, we think it de-risks a lot of the errors that we see with centralized manufacturing of CAR-T products, and hopefully lead to less manufacturing failures and less concerns with getting patients to CAR-T products. So this, I really just wanted to show our whole pipeline. Across BioCanRx, we've built, like I said, plasmid generation and lentivirus manufacturing, all of which is central. But now we've built in clinical trial protocols and patient partners that help build feasible and well-designed trials moving forward, not just for CD19, but for every other tumor target that we plan to test. And we are building this cross-Canada point-of-care sites to really rapidly deploy our Made in Canada CAR-T products across. And so we started as a group of a handful of physicians and scientists across Canada who had an idea in mind. And now with many partners and many funders across the country, we are building a pipeline where we can get from discovery into the healthcare system. Here's the manufacturing. This is what CLIC enabled us to do, was this whole middle piece. But now we're working on new targets starting from scratch where we do all the preclinical testing. And that, if it can feed into the network and the platform that we've built, would hopefully get these to the healthcare system much faster than if we didn't have this platform that we've just built. And so we hope that this middle part, having built this, will now allow us to get from preclinical to clinical much faster and much easier than what we had before for Canadian patients, but ultimately, hopefully, globally as well. Understanding a platform like this can have so many implications for other places that want to do similar things to what we're doing here in Canada. And while I didn't have time to outline that today in my talk, I did want to say that, we are working with other countries who have a vision similar to ours. For example, Denmark has opened a trial using our virus and construct and our clinical trial protocols to build their own clinical trial with our product, and have the same vision in mind of getting more feasibility and more access for their patients to CAR T-cell products. And that's really been a huge excitement for us in getting another country to work with a vision that we have and to have similar plans in mind for their own program. So lastly, I showed you very few people at the beginning, but this program is much bigger than just a few people. Harry Atkins has been one of the original sort of godfathers of this program, and one of my mentors who helped build this program with us, as well as John Bell. And I already mentioned Rob Holt and Brad Nelson. Kevin Hay is my co-lead for the clinical trials now, as Raymond Brody, who I mentioned earlier, has now retired. And I wanted to name 3 important people who I think any program like this really requires. One is Maruna Bala, who essentially is the head of quality for the entire program. And without her, all of what we do from a regulatory side would be almost impossible. So Maruna is really in charge of the quality program, and that is vital to getting a program like this off the ground. Vari Sigrist has been our program manager, who keeps all of our funding and patient partners and trials and deadlines all cohesive and together. And you need a leader like that who can make this program happen. And John Webb and now Julie Nielsen, who runs our T-cell manufacturing and helping with the rollout across Canada, is really pivotal in making sure this all happens. So I really can't thank all of these people enough. And of course, all of our patients who have allowed us to build this network, who have helped us, and who, of course, have put their lives in our trust when enrolling into our clinical trials. So I have to, of course, thank them. And lastly, just really any questions. I thank you for your time, and I am happy to answer questions.
Thank you, Natasha, for that great presentation. We're now going to start our Q&A session. So we'll dive straight in here, Natasha. Our first question here asks, if you reach market authorization for your CD19 CAR, how will this roll out in Canada?
Great, thank you. And sorry about that. So for what we plan to do next for market authorization is really to build an entity who can take on this part after clinical trial. So the standard of care arm that would build this with funders to ensure that we could create a sustainable program for products that we can reach market authorization with. So that's what we're in the process of doing right now, is building that not-for-profit organization who can take on how to roll this out in a clinical way that's not so much the same as what we've done now in a clinical trial. It's very different when we get to standard of care. And so that's kind of what we're working towards now.
Thank you for that answer. So the next question here asks, and I think you did allude to it a bit in your presentation, do you know how this sort of platform is working in other countries?
Yeah, so we've been really excited to see what has happened in Spain. So Spain has a CD19 CAR product similar to ours, manufactured on a Miltenyi Prodigy machine, that they've been able to roll out across the country through hospitals with multiple Prodigy machines across the country, using a hospital exemption pathway to ensure that they don't have to lose time in waiting on the entire EU approval that is needed for products in Europe. And that's going very, very well. We've been excited to hear them speak at meetings and talk about how this has brought access for their patients to a CD19 product in their own country. So we have seen this now starting to pop up in other countries, and I think Spain is probably the furthest along in that regard, but we're very excited to see what other countries are able to come up with as well.
Fantastic. Sounds very exciting indeed. So our next question here asks, now that you are up and running, what is your biggest challenge facing you now?
I think it's sustainability. So we proved that we can do this. We have treated patients, we've cured some, but it is still a lot of money to now build point of care across the country in a sustainable way. Although, of course, we're going to show, hopefully, to our funders that we are a feasible approach to bringing an economical solution to CAR T therapies in this country. That is only the beginning. We actually need money behind that to sustain this program. So I think that remains our biggest challenge. We've had lots of excitement till now, but this part is not as exciting to scientists or the scientific community. So grants are not going to give us money just to stay open. We really now need to talk to our government agencies and funders who can help us sustain this in the long term. And that's the harder part right now, but that's what we're working on.
Fantastic. Thank you for answering that. So this next question again is, I think, looking forward to the future. This member asks, what are the next CAR T clinical targets you are looking at?
So CD19, of course, is well known and be the beginning. We're now working towards targets using llama-derived antibodies, meaning that, you know, we think that could be different. They're single domain antibodies, and that right now is a CD22 target that we hope hope to open in 2024, but we're also looking at targets in solid tumors. And I won't get into the details of which ones we're going to choose next, but we're working towards different targets in different tumors that we think could provide real solutions for certain patients who are failing other therapies.
Fantastic. Thank you for the clarity on that. The next question here asks, with CD19 commercial products now available in Canada, how do you compete?
Yeah, it's a fair question. I asked one of my colleagues in the US when we started and said, is it worth doing a CD19 knowing that they have access to commercial products? We were about to have access. And he's at a very big, prominent center in the US and said, we've got 10 trials at any given time for targets like CD19, because there's always going to be other ways to do this. And so I am not really competing with pharmaceutical products that are on the market. Patients who qualify for pharmaceutical products that are funded by our government go towards those standard of care products. Our trial is for those patients who can't get there, who either don't meet the very strict eligibility criteria that are set up for us for these patients, or who are just too sick to wait the 4 to 6 weeks. And so a handful of our patients, many of them have these diseases that wouldn't qualify for standard of care or who are too sick. So it is not really a competition right now for us in terms of who's coming to the trial.
Fantastic. Thank you for the clarity on that. Our next audience member here begins by thanking you for an excellent presentation. And then they said, in your opinion, what are the critical factors that need to be addressed to move point of care CAR-T care from trial format into ongoing routine clinical practice?
That's a tough question. In Canada, we really need to work towards a regulatory pathway for these therapies, and Health Canada is working with us on that. We've seen this now in the UK and in Europe in terms of not just the hospital exemption pathway, but a regulatory path for these products that are not the same as centrally manufactured, pharmaceutically driven products. They are different. And so that's part of it, is working with our regulatory body to bring this to the market, but then also working with our funders to ensure that once we get to a market authorization platform, that we have a way to sustain this and fund this in the long term so that we can continue to provide access across the country.
Fantastic. Thank you for answering that. The next question here asks, could you please provide more information on your lab clean room to be able to manufacture CAR-T at the hospital?
That's great. So actually, we don't manufacture in the hospital building itself. It's manufactured in our research institute. And the classification right now is essentially a grade D clean room. And what that needs to look like when we're at the stage of market authorization or outside of clinical trial is still to be determined. But right now, we function in a grade D clean room.
Fantastic. So thank you for answering all those questions. And as to our audience, we thank you for listening and we hope you'll join us again soon.